the ship first sank.
A shipwreck is a huge example of human waste. But what about the little stuff, like plastic?
Our planet is facing a big problem. Plastic pollution that clogs our waterways and harms our precious marine life. But we're researching a solution that could make a huge change. Instead of fossil fuels, bioplastics are made from natural materials like sugarcane. We are testing a specific kind called polylactic acid, or PLA. It's used in 3D printers to create almost any shape, so it's a really versatile material. In our labs, we're studying which PLAs break down best and what size they break down to. We do this in our chemistry lab with bacteria. The bacteria eat the organic matter in the bioplastic and break it down. This means lots of very small particles end up in the fluid. But we also want to know how bioplastics break down in the real world. To do that, we need to print some samples. Our 3D printers use 5 kinds of PLA. One is pure, the rest mix sugarcane with materials like algae, bamboo, or cherry wood. We put small samples into balls with holes so that water can flow through. These balls go into a waterway where bacteria in the water begin to eat the bioplastic. After 18 months of this, we can bring the samples back to our lab for testing. For this, we use a technique called infrared spectroscopy. This technique bounces infrared light onto our sample to measure any changes. The PLA absorbs unique wavelengths of light and shows these as peaks on a graph. These results can tell us if bacteria have broken down the surface of the plastic. We need to find out which bacteria like eating bioplastics. This requires some DNA testing. First, we isolate the DNA of the bacteria. This is done in a highly sterile environment. Any contamination could affect the result. We use liquid enzymes to separate DNA from the membranes of the bacterial cell. Then the DNA is sent off-site for a very specialized test called DNA sequencing. When the results are in, we can identify the exact bacteria we found on our 5 PLA samples. Then, we can match that bacteria to the sample that degraded fastest. That gives us clues about which organic materials are the most environmentally friendly.
Did you know there's a highway for humpbacks? And I'm not talking about whales driving.